After studying this module, you shall be able to have the structural insights of DNA, know about the chemical properties of DNA and RNA, learn about the various forms of DNA, and know about the denaturation and renaturation or the reannealing of DNA, which forms the basis of many molecular biology techniques. In the previous module, we have learnt about the composition of nucleic acids. DNA comprises of deoxyribonucleotides containing bases adenine, guanine, cytosine and thymine. Remember, uracil is absent in DNA. While RNA comprises of ribonucleotides with uracil instead of thymine, and adenine, cytosine and guanine are present the way they are present in DNA. RNA usually exists in a single-stranded form, while DNA exists in a double-stranded form. In double-stranded DNA, adenine always pairs with thymine, A-T bond, and guanine always pairs with cytosine, which is a GC bond. DNA exists in different forms inside a cell. They are A form, B form or Z form, which are defined by different humidity conditions. Now, looking at the base composition of nucleic acids, let's study about DNA first. So, when we say deoxyribonucleic acid, it contains two polydeoxyribonucleotide chains running in antipolar or the opposite directions, such that one strand is in 5' to 3' orientation and the other strand is in 3' to 5' orientation. Hence, DNA is double-stranded. These two strands coiled around a common axis, forming a structure which is resembling to a spiral staircase. This is known as a DNA double helix. The two strands of the double helix are held together by interchain hydrogen bonds. The bases in one strand base pair with the bases in the other strand. This is termed as base pairing. Remember, these are the hydrogen bonds. The information coded in the DNA can be assessed through major and minor grooves. Now let's look at how the structure looks like, structure of DNA. This is one polynucleotide chain, this is another deoxyribonucleotide chain. Now the adenine from one chain projecting outward from one of the chains base pairs with thymine, and a G projecting from one of the chains base pair with the cytosine of the other. This is termed as base pairing. Now, looking at this particular figure, we can see the minor and the major grooves. Major grooves, which is larger in size compared to the minor groove. This is the region where proteins assess the DNA. Now let's move on to the Chargaff rules. Erwin Chargaff analyzed the base composition of various DNAs and found that 4 bases of DNA do not occur in equimolar proportions, but it is the other way around. It is adenine and thymine are found in 1:1 ratio. Also, guanine and cytosine are also found in 1:1 ratio. The data shown in table is basically what Chargaff saw. Second column shows %A, third column shows %G, %C, %T. Our A is to T ratio comes out to be nearly 1 in various organisms, while our G is to C ratio also comes out to be nearly 1 in most of the organisms. Thus, it means that the concentration of A equals T, also concentration of G equals C. If we add both, A plus G equals T plus C. Thus, purines equal pyrimidines in case of DNA. These are called the Chargaff rules. Now let's learn about Watson and Crick base pairing. Two scientists, James Watson and Francis Crick, worked on the structure of DNA. Rules of Chargaff and also data from the work of Rosalind Franklin and Maurice Wilkins helped them to decipher that the two strands of DNA are held together by hydrogen bonding between purine on one strand and pyrimidine on the other strand. Adenine base pairs with thymine while guanine base pairs with cytosine, as we just learnt. An AT base pair is held by two hydrogen bonds while a GC base pair has 3 hydrogen bonds. Thus, if one knows the sequence of one strand governed by the rules of complementarity, we can also predict or know the sequence of the other strand. For example, if a strand has a sequence A, T, 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 A, A, G, 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 C, G, A, G, C, G, A. By convention, the sequences are always written from 5' to 3' direction until and unless it is specified. So, by the rules of complementarity, T should face A. So, the sequence becomes, the sequence of the complementary strand will be TAA, AAA, TT, CC, CGC, TC, GCT. Remember, the DNA strands are in anti-polar orientation. So, if the first strand which was given to you is in 5' to 3' direction, the complementary strand we just wrote has to be in 3' to 5' orientation. This was about DNA. Let's now move on to the other nucleic acid which is found in the cell, which is ribonucleic acid or the RNA. RNA, however, exists in a single-stranded form. Although there are exceptions to this rule. There are viruses that contain single-stranded DNA or double-stranded RNA as their genome. RNA comprises of a single polyribonucleotide chain with A, G, C and U. Remember, there is no thymine in RNA. However, unusually certain bases are found in few forms of RNA which can be methylated bases, inosine, thymine to a lesser extent, dihydrouridine and pseudouridine. In a cell, RNA exists in 3 predominant forms. Messenger RNA. As the name suggests, this RNA is the messenger that carries the information of the DNA to protein synthesis machinery. Thus, it is acting as an intermediate between DNA and the protein. Protein synthesis machinery translates this information into sequence of polypeptides. The second RNA which is found in the cell is transfer RNA or tRNA. It is the smallest RNA found in the cell, comprising only of 76 nucleotides. Transfer RNA serves as a carrier of amino acids, which are the monomers for protein synthesis. Thus, it is also single-stranded, but it assumes or forms a characteristic secondary structure by virtue of intra-strand hydrogen bonding. The third form of RNA which is found in the cell is the ribosomal RNA or the rRNA. rRNA forms the part of the ribosomes. Ribosomes, the protein synthesis machinery, comprises of 1/3 proteins and 2/3 RNA. Now this 2/3 RNA is the rRNA. rRNA is the most abundant RNA which is found in the cell. They participate in the formation of peptide bonds that link any two amino acids in a protein chain. Hence, they are called ribozymes or RNA enzymes. just now learnt about DNA as well as the RNA which is found in the cell. DNA can exist in various different forms, namely bDNA, aDNA and zDNA. This is a figure which shows the structural comparison of these A, B and Z DNA. bDNA is the one which was deciphered by Watson and Crick. Both these strands are in right-handed turn. This is formed under conditions of very high humidity and thus corresponds to the structure of DNA under physiological conditions. It has around 10 base pairs per turn. It has wide major groove and a narrow minor groove. Other form of DNA which exists in the cell is the aDNA. This form is observed under conditions of low humidity. It has around 11 base pairs per turn. Its major groove is narrower than the bDNA. The various characteristics of the A form or the B form are listed in this particular table. Given the A form has a right-handed sense, B form has a right-handed sense, while the Z form has a left-handed sense. We are going to shortly learn about Z form also. The Z DNA. When our DNA contains alternating purines and pyrimidines, it assumes a left-handed conformation. While the glycosidic bonds connecting the base and the sugar are in anti-conformation in right-handed helices, they are in anti-conformation for pyrimidine residues and in syn conformation at pyrimidine residues. This alternating pyrimidines and purines in anti or syn conformation gives it a zigzag pattern and hence the name zDNA. It accommodates around 12 base pairs per turn. Now this is with the various forms of DNA which are found in the cell. Remember in the last module and also in this module we learnt that DNA and RNA differ from each other majorly by two differences. There is presence of uracil in RNA versus thymine in DNA and the presence of 2'-deoxyribose in DNA while RNA contains the ribose. Major consequences on their properties. These chemical properties render stability of DNA, making it most stable form of genetic information. 2'-OH of ribose in RNA makes it less stable than DNA. The vicinal hydroxyl group next to 3'-OH, that is which is the 2'-OH, is absent in DNA, rendering it more resistant to hydrolysis by alkali. This vicinal hydroxyl group in RNA makes the phosphodiester bond susceptible to nucleophilic cleavage. Now let's ask a question. Why does DNA contain thymine instead of uracil at the first place? Cytosine deaminates to uracil in vivo. Cytosine base pairs with G, while uracil base pairs with adenine. Thus, this deamination leads to a mutation, leading to conversion of CG base pair to a UA base pair. Thus, U in DNA is an outcome of deamination and is sensed as a mutation by the repair machinery. If U, which is uracil, would have been naturally found in DNA, the repair system of the cell would have been unable to sense uracil formed from deamination of C. The two complementary strands which are present in DNA, which are held by hydrogen bonds, cannot be easily brought apart. When a solution of DNA is heated to temperatures above physiological temperatures or under conditions of extreme pH, the two strands of DNA come apart, a process which is termed as denaturation. However, it's a reversible process. When the denatured solution of DNA is gradually cooled, the two single strands start looking for their complementary zones in their partners, and hence they locate them, they renature by forming the hydrogen bonds. This property is also used in re-annealing. Re-annealing permits the formation of hybrids when the two strands are of different origin. This is also termed as hybridization, that forms the basis of many techniques in molecular biology, such as gene expression microarray and chip-on-chip. DNA maximally absorbs UV light at a wavelength of 260 nanometer. When the solution of DNA is heated above near to the boiling temperatures of water, absorbance at 260 nanometer increases markedly. There is approximately a 40% increase. This is termed as hyperchromicity. This is an outcome of diminished base stacking interactions as due to opening up of the double helix. If we can look at in this particular figure, the increase in absorbance at 260 nanometer, when it is plotted against the temperature, a sharp curve is obtained. The midpoint of the rise in absorbance marked the melting point, or also known as the TM. This melting temperature is higher in DNA, which is rich in GC content, because more energy is required to break 3 hydrogen bonds of GC pair compared with 2 hydrogen bonds of a AT base pair. Look at this figure. When denatured, the 2 strands of the DNA come apart. This is a partially denatured DNA which is shown. The complete separation is going to lead to the complete dissociation of the 2 strands. This is the melting curve, in which this is with no denaturation. At the TM point, which is the melting temperature, half of the DNA is denatured, half of the molecule opens up. And 100% denaturation is something of this sort, in which 2 strands come apart and there are no hydrogen bonds which link one strand to the other strand. And remember, we also learnt about that higher the GC content in our DNA, the higher is the TM, as depicted by the graph in this particular figure. To summarize, DNA is double-stranded with inter-strand hydrogen bonding between adenine and thymine pair and guanine and cytosine pair that keeps the 2 strands together. DNA exists in 3 predominant forms in the cell: A, B and Z form of DNA. The 2' OH of ribose in RNA renders it unstable as compared to DNA. The complementarity between the 2 strands of DNA forms the basis of renaturation or re-annealing or hybridization utilized in many molecular biology applications such as microarray.